Okay, and we'll start sharing my screen.
Just a notification, it's recording, okay?
Yeah, it's recording now. Okay, so can you see my screen right now?
Yep.
Okay. Sorry,
I'm just
give me a
gonna
moment.
I'm just gonna mute myself.
Okay. Okay, so as you probably already know, my name is Ashley. Thank you for coming. So I'll be leading today's topic session, which is about metabolism, part one, and this is a huge topic on the biochemistry section for the MCAT. So I broke down the metabolism section into three parts: anaerobic respiration, aerobic respiration, and metabolism. But today we will be focusing on glycolysis, fermentation, and the regulation between gluconeogenesis and glycolysis. So just a warning, we'll be going through each of the 10 steps of glycolysis. So it's about to get really in depth. So make sure to take notes to label the starting materials, the enzymes, and the products and byproducts as well, because you will be asked questions about them. So starting with glycolysis, it is located in the cell's cytoplasm. It occurs in eukaryotes and prokaryotes. It is anaerobic respiration because it does not involve oxygen. And glycolysis is notorious for producing energy very quickly. So how can we relate cancer cells to glycolysis? Is there something unique about cancer cells?
Their replication rate.
Yeah, so cancer cells will reproduce very quickly. They will undergo cell replication, and they're going to need a lot of energy to do this. So they're going to heavily rely on glycolysis to pump out a lot of ATP very quickly, and they'll bypass aerobic respiration so that they don't have to go through all of the steps of the Krebs cycle and the electron transfer chain. So for them, it's much easier to just go straight with glycolysis and rapidly produce those 2 ATPs per glucose. So these are the 10 steps of glycolysis. Do not be intimidated by them right now because we will go step by step. So it's really easy to memorize all the 10 steps of glycolysis. Like if you really focus on memorizing it, you can do it. But it is a waste of time. And the best way to understand glycolysis is to understand what the enzymes are and to break down the function of the enzymes by looking at the names. So kinases, as we know, is responsible for adding phosphate groups to molecules. Isomerases are responsible for rearranging the molecule. Aldolases break down molecules. Dehydrogenases, they are a type of oxidoreductase which perform oxidation and reduction reactions. But dehydrogenase, as the name suggests, dehydrogen, it removes a hydrogen atom. And finally, mutases are just a type of isomerase, so it just also rearranges the molecule. So step one of glycolysis is really important. It is irreversible, and it involves the enzyme hexokinase. So looking at the name hexokinase, hex means 6, kinase, as we just said, adds a phosphate group. So we have a 6-carbon sugar, glucose, and we are going to phosphorylate it on the 6th carbon, giving us glucose 6-phosphate. So we're snatching that phosphate from ATP, and we're making ADP and glucose 6-phosphate. So why would the cell want to phosphorylate glucose in the first place? Why is this important?
To enter the internal mitochondria, I think where the cellular reactions occur.
Okay, so glycolysis, it occurs in the cytoplasm of the cell, like it doesn't involve the mitochondria. So it's important to phosphorylate glucose because if you add that negative molecule in pink, the phosphate group, by attaching it to glucose, it will be so much harder for glucose to exit the membrane. So by phosphorylating it, you're trapping glucose into the cell. So once you've trapped glucose in the cell, it'll be easier for it to undergo glycolysis. So this step just basically traps glucose so that glycolysis can happen. Does that make sense?
Yes. So then it becomes pyruvate, right? Just to make sure. So
yes,
that it becomes
that's the end product.
Smaller molecule to enter the. Yeah. Okay. Gotcha.
Yeah. Okay, I was just double checking if I'm recording. Okay, so step 2 of glycolysis involves phosphoglucose isomerase. So looking at the name of this enzyme again, we have a phosphorylated glucose and isomerase. So we're isomerizing this glucose 6-phosphate into fructose 6-phosphate. And this can be seen by looking at this 6-carbon sugar, and we're converting it into a 5-carbon sugar. So fructose 6-phosphate is an isomer of glucose 6-phosphate. So moving on to step 3, we have fructose 6-phosphate, and we have the enzyme phosphofructokinase 1. So this step of glycolysis is really important as well because it's irreversible. So step 3 is the commitment step, and it involves PFK1, which is the rate-limiting enzyme. So looking at PFK1, we have a phosphorylated fructose kinase 1. So we're phosphorylating this phosphorylated fructose on the 1 carbon, right? So we have this fructose, it has the phosphate group on the 6 carbon, and we're adding it to the 1 carbon. So once again, we're snatching that ATP, making ADP. So this is still part of the investment phase when we're investing ATP so that we can have a net gain of ATP. Okay, so step 4 of glycolysis. Now that we've made our fructose 1,6-bisphosphate, we're going to use aldolase to break down the enzyme. As I said, aldolase is responsible for breaking molecules. But the problem with this step is that it breaks fructose 1,6-bisphosphate into 2 uneven products. Our goal here is to make 2 molecules of glyceraldehyde 3-phosphate. But like I said, because we have 2 uneven products, we're going to make dihydroxyacetone phosphate. So we're going to use triose phosphate isomerase to convert DHAP, which is a short name for dihydroxyacetone phosphate, into glyceraldehyde 3-phosphate. That's all we're doing. So looking at this enzyme, triose phosphate isomerase, we have this 3-carbon sugar that's phosphorylated, as we can see here on this 1 carbon, and we're isomerizing it to make G3P. So that's the end goal for this step, to make 2 molecules of G3P so that we can keep going through glycolysis. Does that make sense? Okay. So step 6 now. Now that we have our 2 molecules of G3P, we got what we wanted. We have the enzyme glyceraldehyde 3-phosphate dehydrogenase. So like I said before, dehydrogenase is a type of oxidoreductase. And we know that we're going to have an oxidation and a reduction reaction. So G3P is going to be converted into 1,3-bisphosphoglycerate. So the way we can see this is that we are adding a phosphate group onto G3P on the 1 carbon. So we're replacing this hydrogen with a phosphate. So G3P is being oxidized because this hydrogen is being removed by the dehydrogenase, right? So once we snatched the hydrogen out from G3P, we need something to accept it because we can't just have hydrogen floating around. So this hydrogen will be added on to NAD+, and an inorganic phosphate that's floating around the cell will replace that hydrogen. So NAD+ is being reduced to NADH. It's really, really, really important to have NAD+ available so that this step can occur. Because without NAD+, we won't be able to replace this hydrogen on G3P, and we won't be able to add a phosphate to it, and then glycolysis will just stop. So also keep in mind that step 6, 7, 8, 9, and 10 will occur not twice, but like, you know how we started with glucose, the 6 carbons? We've broken it down to glyceraldehyde 3-phosphate, which is a 3-carbon molecule, and there's 2 molecules of that. So every starting material, byproduct, and product will have 2 versions of it. So that's why it says in parentheses 2 molecules, 2 NADH, 2 NAD+, and 2 NADH.
Ashley, I had a quick question.
Yeah.
So where does the FADH come into place?
NAD plus or FADH?
The FAD.
Okay, so FADH2 is not involved in glycolysis. You'll see more of that in the Krebs cycle and on the electron transfer chain, but we're not covering those reactions today. We're just focusing on glycolysis.
Just on the glycogen. Okay, gotcha.
Yeah.
Thank you.
So step 7 of, yeah, no problem. So step 7 of glycolysis, now that we have our 1,3-bisphosphoglycerate, we have our 2 phosphates on our glycerate, we have the enzyme phosphoglycerate kinase. So this glycerol is phosphorylated and we have this kinase, but unlike the other steps, we're not phosphorylating our starting material, but rather we're phosphorylating ADP. And as I guess showed with this little cartoon on the upper right-hand corner, this is an ATP step. So we're finally in the payoff phase. We're finally making ATP, but this 2 ATP is still not enough to have a net gain. We'll see where we're gonna have our net gain of ATP later. But the main idea is that we are finally making 2 ATP by snatching this phosphate group on the 1 carbon of 1,3-bisphosphoglycerate, and we're adding it onto ADP. So now we're just left with 3-phosphoglycerate, right? So you can see, like, by the name change, 1,3-bisphospho, we just have 3-phospho. Okay, so now step 8. This is relatively simple. So we have our 3-phosphoglycerate and we have phosphoglyceromutase, the enzyme. So mutase, like I said before, is a type of isomerase. So it's going to rearrange the molecule. And as we can see, we have our phosphate group on this carbon, and it is going to swap places with alcohol. So that's all we're doing with this step.
Ashley, are there any
Yes.
electron transfers or proton transfers?
No, not for this step.
It's just swapping, right?
Yeah, just swapping.
Okay, thank you.
So step 9. Now that we have our 2-phosphoglycerate, we have our enzyme enolase. And that is going to convert 2-phosphoglycerate to phosphoenolpyruvate. So enolase is an enzyme that's responsible for creating carbon-carbon double bonds. So phosphoenolpyruvate, this enol here is telling you that we have a carbon-carbon double bond. So that's all it's doing. And to make that, it's going to secrete 2 H2Os. So enolase has nothing to do with phosphate, has nothing to do with dehydrogenases. It's just making this carbon-carbon double bond. And finally, we have another very important step, which is step 10 of glycolysis. It is an ATP step. So we have our phosphoenolpyruvate and pyruvate kinase. So we're finally making our 2 ATP net gain. So like I said before, this kinase is unusual from the investment phase because we are taking the phosphate from phosphoenolpyruvate and we're attaching it to ADP to make ATP. So we're just left with pyruvate. And as you can see, like I said before, enol means carbon-carbon double bond. That is no longer present in pyruvate. And step 10 of glycolysis is also a commitment step. So as you can see, this is irreversible. And it's important for this reaction to be irreversible because it's the last step. And there were what, like 10 steps before, or I guess 9 steps before. So the cell wants to make sure that glycolysis will be finished and that we're going to get our two pyruvates. So that's why it will make this reaction irreversible.
Ashley, sorry for asking
Yes.
so many questions.
No,
So
it's
over
okay.
here, you said that the phosphate's gonna go, and you're gonna have like a ketone structure, right?
Yes.
But like, where did does the double bond go to the oxygen by tautomerization?
Uh, um, yes. Yeah, okay. So let me see here.
Yeah, so the double bond
So
goes over there, and the phosphate like leaves. I think that's tautom. I think so. Yeah.
yeah, it is. The MCAT won't ask you like that in depth, like the
Okay.
mechanism of glycolysis. It's just important to know like the enzymes and the products.
Okay. Thank you so much.
Mhm. So this is finally the overview of glycolysis. We have all of our 10 steps laid out, and I hope now it's a little bit more easier to see, not so intimidating. Okay, and I want to ask you a few questions about glycolysis. So which of the following molecules are not products of glycolysis? I'll give you, I guess, 2 minutes to answer this.
Carbon dioxide.
Yeah. Okay, that was a lot quicker than 2 minutes. Good job.
Thanks so
So,
much.
how did you get to carbon dioxide?
Like I was like a bit sure it was carbon dioxide, but I took some time because I was like looking around for carbon dioxide. It wasn't even inside the reaction, right?
No, it
I
wasn't.
couldn't find it. Yep, it
Yeah.
wasn't in there. I just
So,
wanted to confirm. That's why I took some time.
yeah, so the MCAT will sometimes, or I guess a lot of the times, they will throw a lot of molecules into answer choices, so to get you a little bit confused and intimidated. But um, like once you understand what they're asking you and the concept, and you just think it through, then it's easy to see that like a question like this could be a trap.
Thanks so much.
So this is my reasoning behind it. And if you don't remember, I'm sorry, could you please mute yourself?
Oh, I'm so sorry.
There's a lot of background noise. Sorry, that's okay. Thank you. So to walk you through this question, I know that you got it right, but like to eliminate the other wrong answers. So if you didn't know the starting materials and the products of glycolysis, you can easily choose which ones to eliminate. So starting with the ATP is of course a product of glycolysis because that's the purpose of glycolysis to pump out ATP. So pyruvate, if you remember, glycolysis means breaking down glucose. So you have that 1,6 carbon sugar, and we're breaking it down into 2, 3 carbon sugars. And those 3 carbon sugars are pyruvate. So if you're torn between NADH and CO2, you can remember that in the step of glycolysis that involved the dehydrogenase, we're snatching that hydrogen from a carbon and we're giving it to NAD+. If you couldn't remember that, then you can also remember that glycolysis also generates electron donors, or I guess electron carriers to the electron transport chain. So those electron carriers are NADH. So that's another like function of glycolysis is to make those electron carriers for the electron transport chain, which will generate a lot more ATP. So moving on to the next question, what is a product of the first commitment step of glycolysis?
Glucose 6 phosphate.
So this is sort of a difficult question. So what I meant by the first commitment step of glycolysis is the first commitment step that's unique to glycolysis, which is
Or did you mean like the whole reaction or just the first step?
the whole reaction.
Oh, the whole. If it's the whole reaction, I'm thinking of pyruvate.
Okay, so pyruvate is actually the product of the second commitment step of glycolysis.
Okay.
So as I said before, there's three irreversible reactions, but step 3 of glycolysis is the first commitment step, which involves phosphofructokinase 1. So once you start getting familiar with glycolysis, you'll recognize that phosphofructokinase 1 is a really important commitment step of glycolysis. And you can remember the product that this enzyme makes by looking at the name of the enzyme. So phosphofructokinase 1. You have a phosphorylated fructose, and you're going to phosphorylate that on the 1 carbon, making fructose 1,6 bisphosphate. So this is a little explanation that I wrote. So next question, which of the following reactions in glycolysis produces ATP?
B because remember that cartoon?
Oh, sorry. Yeah, you're right. So let's break it down. So number one, glucose to glucose 6-phosphate. This is the first step of glycolysis, which is still in the investment phase. So you're not going to be making any ATP because it's not the payoff phase. So A and C are immediately out. So two is automatically correct because they're the two answer choices left. So you really have to just see if three is correct. So 2-phosphoglycerate to phosphoenol pyruvate. So like I said before, enol here is telling you, it's giving you a hint so that there's a carbon-carbon double bond. And if we remember the enzyme that makes this double bond, it'll be enolase. And like I
Yep.
said before, enolase has nothing to do with phosphate, has nothing to do with um, NADH. It's just creating that double bond. So since it has nothing to do with producing ATP, we can eliminate that, and B is our answer. Okay. So now we'll move on to gluconeogenesis. And breaking down the name of this, it means creation of glucose. So this process occurs in the liver cells. It happens when there's limited glucose. So when we've been starving for a long time and our blood sugars are very low, and our cells will begin gluconeogenesis to correct this low blood sugar level. And gluconeogenesis, unlike glycolysis, has 11 steps. It's referred to as reverse glycolysis because it still deals with the same molecules, it's just different enzymes. Oh, I'm sorry. So be familiar with the overview. You don't have to memorize like every single byproduct. Just be familiar with these 4 enzymes in blue and take note of this equation. So you can just notice that there are so many starting materials that go into making just one molecule of glucose. So it's telling you that because there's so many things going into this process just to make one 6-carbon sugar, that it's very important. And so this molecule has to be heavily regulated so that the cell isn't undergoing futile cycling, so that it's not competing with glycolysis. So these are the reciprocal reactions that control glycolysis and gluconeogenesis. So like I said before, because gluconeogenesis has so many things going into it, the cell wants to make sure that there are control points that regulate like if glycolysis is happening and gluconeogenesis stops or vice versa. So these two processes cannot happen at the same time. That would be very damaging, or I guess it would just be pointless and you're just wasting so many resources. So the first one here is
It's just
hexokinase.
sorry for
Yeah.
interrupting. It's just gonna be
Mhm.
neutral, right? Because if you go one way and the other way, it's just gonna cancel out, and you're not gonna have anything.
Yeah, so it either goes up or down. That's it.
Okay.
Yeah, so the first one is hexokinase. So, like I said before, it phosphorylates glucose on the 6th carbon. So, gluconeogenesis will do the opposite. It takes glucose 6 phosphate and it removes the phosphate on the 6th carbon. So, it uses phosphatase to use that, to do that. And the next one, phosphofructokinase 1, like I said before, this is the first commitment step of glycolysis, really important, heavily regulated. So we have fructose 6-phosphate, and we're making fructose 1,6-bisphosphate for glycolysis. Gluconeogenesis will do the reverse. It will take that phosphate on the first carbon of fructose, and it will make fructose 6-phosphate. If that makes sense. Okay. And finally, we have our last step of glycolysis, pyruvate kinase. So you have phosphoenol pyruvate making pyruvate. And gluconeogenesis, like I said before, it involves 11 steps. This is the extra step that is not seen in glycolysis. So you have pyruvate, and you have to convert it into oxaloacetate using pyruvate carboxylase. And you'll use the enzyme phosphoenol. I forgot the full name of it, but I usually refer to it
I'm
as
just guessing.
PEP.
I'm just
Yeah,
guessing, but is it phosphoenol pyruvase? I'm just guessing.
that's what I was thinking. Let me see. I'm sorry. There's so many names for this. Yeah, so it's phosphoenolpyruvate carboxylase. I just forgot to write carboxylase to make phosphoenolpyruvate. So the purpose of this is to convert pyruvate to phosphoenolpyruvate. It just requires this extra step. Okay. So regulation of glycolysis and gluconeogenesis. There are two heavily regulated enzymes. So like I said before, the first commitment step of glycolysis involves phosphofructokinase 1. And the reciprocal of that for gluconeogenesis is fructose 1,6-bisphosphatase. Yes. So in a low energy state, AMP will activate PFK and it will inhibit fructose 1,6-bisphosphatase. So AMP, I guess high levels of AMP will tell you that the cell is starving of energy. So if you have high AMP, you will have low ATP. And I think I wrote down, right. So AMP stands for adenosine monophosphate and ATP stands for adenosine triphosphate. So AMP is kind of like the opposite of ATP. So if you have high levels of AMP, that means the cell has low energy. So it will activate phosphofructokinase 1, the commitment step of glycolysis. So it will encourage glycolysis to be committed. So fructose 6-phosphate will be converted into fructose 1,6-bisphosphate, and glycolysis will continue. But at the same time, AMP will inhibit fructose 1,6-bisphosphatase. So it's encouraging glycolysis to continue, and it's stopping gluconeogenesis from happening. Does that make sense? Okay. So this is another type of regulation of glycolysis and gluconeogenesis. So fructose 2,6-phosphate is a molecule that we haven't talked about yet. And it regulates PFK, phosphofructokinase, and fructose 1,6-bisphosphatase. So this molecule is regulated by the hormones insulin and glucagon that are produced by the pancreas. So if you have high blood glucose, so after a good meal, we have lots of insulin. This insulin will activate the molecule fructose 2,6-bisphosphate. This molecule will activate PFK. And as we know, PFK is the committing enzyme of glycolysis. So it will encourage glycolysis to continue. And this makes sense. So like if you come across questions like these on the MCAT, it's really helpful to walk through it like a story. So after eating a meal, your insulin levels are high, and this will tell you to use up the ATP from your food. Use up the glucose from your food to make energy, or you can store it as glycogen, but that's a different story. So in
And
the opposite case, sorry.
sorry, uh, gluconeogenesis would be the other way around, right?
Right. So if we have low blood glucose, if we've been starving for a while, our blood glucose levels are low, our glucagon levels will be high to tell our body that we have low blood sugar. So glucagon, unlike insulin, it will inhibit fructose 2,6-bisphosphate. And if this molecule is inhibited, it will activate fructose 1,6-bisphosphatase, which is the reciprocal committing enzyme of gluconeogenesis. Let me see if I can. Right. It will inhibit this. So it kind of acts like AMP because it will inhibit fructose. I'm sorry. No, it will activate fructose 1,6-bisphosphate. So forget what I just said in the last minute. So glucagon will inhibit fructose 2,6-bisphosphate. And
And F16 bisphosphate will like inhibit the other one, the gluconeogenesis, right?
yeah, so basically fructose 2,6-bisphosphate will encourage glycolysis to continue. So if that molecule is activated, it will activate glycolysis and it will inhibit gluconeogenesis. So if glucagon is inhibiting this molecule, you're releasing that inhibition on fructose 1,6-bisphosphatase. So because this molecule is inhibited, gluconeogenesis is allowed to continue. Does that make sense? I know this part is confusing. Okay, thank you. So this is a summary of glycolysis and gluconeogenesis. This is just a diagram that I wrote. I don't know if it's helpful or not, but this is the way that I studied. So, like I said, insulin will activate fructose 2,6 bisphosphate, and it will activate phosphofructokinase 1. And that will allow glycolysis to continue so that you can make ATP and break the glucose from the food that you just ate. Glucagon will do the opposite. It will inhibit fructose 2,6 bisphosphate, and this inhibition will inhibit glycolysis, but it will activate gluconeogenesis so that you can make glucose. Does that make sense? Is this diagram confusing?
Only the part where you have the plus sign and the minus sign. That doesn't mean you're adding and removing the like insulin and glucagon.
Oh no, the plus and the minus means activate and inhibit. So
Activity.
insulin
Okay.
activates, yeah. So insulin activates this molecule, glucagon inhibits this molecule.
Okay.
And this inhibition will activate this enzyme, so it's free to go.
Okay. Thank you so much.
So now we have some practice questions. Which of the following is true in regards to the regulation of gluconeogenesis and glycolysis?
I'm thinking of A or C.
Okay, so A is wrong because, like I said, gluconeogenesis relies on the inhibition of fructose
Inhibition. Yeah.
2,6.
Yeah. Yeah. I was I
Right.
was. But did we mention acetyl CoA?
Oh, no, we didn't. I just wanted you to see like why A, B, and D would be wrong. So
Okay.
B, the control of glycolysis is indirectly linked to the control of gluconeogenesis, right? So it's either
Yeah.
one or the other. It's not direct.
Yeah. That one is false. Yeah.
So, right. And the majority of enzymes responsible for the regulation of gluconeogenesis are actually found in the cytoplasm, not the
Cytoplasm. Yep.
mitochondria.
Yep.
And let's talk about C. So acetyl-CoA activates enzymes of gluconeogenesis and has an inhibitory role in gluconeogenesis. So acetyl-CoA, if you have high levels of this molecule, it means that the cell has a lot of energy because, although we haven't talked about it yet, but if you have a lot of pyruvate, that pyruvate will be converted into acetyl-CoA, and the acetyl-CoA will be fed into the Krebs cycle to make lots of ATP. So if you have lots of acetyl-CoA, that means you have lots of ATP. So acetyl-CoA will activate, so because you have a lot of ATP, acetyl-CoA will activate enzymes for gluconeogenesis because you have a lot of energy. You're going to invest that energy to make more glucose. And it will have an inhibitory role in gluconeogenesis. I know that doesn't really make sense yet because we haven't talked about the Krebs cycle yet, but the point was to just see like why A, B, and D are wrong. Okay.
I understood it. Yeah.
Okay, thanks.
Thank you.
So a basketball player is doing a high intensity workout. Which of the following processes is likely to be occurring? Is least likely to be occurring?
So glycolysis is occurring. I don't. I'm sorry, but I think I forgot whether you mentioned glycogenolysis, but for sure the answer is D, glyconeogenesis, because it's the other way around.
Yeah, that's exactly right. So we haven't talked about lactic acid fermentation yet or glycogenolysis. I just wanted you to see that gluconeogenesis would not be happening when someone is exercising, because when someone's exercising, they want to make more ATP so that they can continue using their muscles. And gluconeogenesis will just take that ATP to make glucose, and that's not necessary when you're doing a workout. And I said this before, gluconeogenesis happens when you've been starving for a while and you want to make glucose.
And the lactic acid is when you have I think when we learn about like the cell reactions, the lactic acid is when you have like muscle strain. That's because when you're like working out and it's going to become lower and your muscle is going to be like it will start producing lactate or something like that to cause muscle strain.
Yeah, so we'll talk about that like right now.
Okay.
So let's talk about fermentation. So the purpose of fermentation, the primary purpose of fermentation is to regenerate NAD plus for glycolysis to continue during anaerobic respiration. So pyruvate acts as an electron acceptor in fermentation reactions in yeast and humans. Although in yeast it's actually a bit different. The pyruvate has to be decarboxylated into acetaldehyde, and acetaldehyde will act as the electron acceptor. So what I mean by that is pyruvate will accept, I'm talking about in the human reaction, by the way. So pyruvate will accept this hydrogen from NADH, and it will be converted into lactate to make NAD+. This NAD+ will then be fed into glycolysis. So like I said before, I don't remember what step number this was, but the step involving dehydrogenase. Remember how I said that glycolysis cannot continue unless you have that NAD+ to accept that hydrogen? So in order for glycolysis to continue, we're going to have to regenerate NAD+, and that's where fermentation comes in handy. So what happens to the built up lactate in our muscles? You sort of answered this, so I'll just add on to it. So the built up lactate in our muscles will actually be transported to the liver, where it will be broken down into pyruvate, and it will make NADH. And this NADH will be useful for the electron transport chain to make more ATP. So was that too quick? Are you ready for a question?
I just wanted to say that the NAD plus NADH, I was just looking backwards. I think it's on step 6. Yeah, I was thinking of step 7, but it's just before that. Yeah, step 6. Just wanted to mention
Yeah,
that one.
yeah, let me double check. Step 6. Yeah, that's exactly right. Step 6, dehydrogenase. Yeah, so you can see this NAD plus here that we're making. Okay, so this is our final question for today. Which of the following correctly describes the function of fermentation?
We just talked about this. This is produce NAD plus.
Yeah, that's exactly right.
Yep. I think you just mentioned it right, like before you ended, because it's the pyruvate takes the H from the NADH and produces NAD.
Yeah, that's good. I really wanted to emphasize this fact because the MCAT will allude to this function of fermentation a lot, so it's important that you know what it's used for and why it's helpful for the cell. Okay, and that's all of the content and questions that I have for today. Tomorrow I'll be working with Sim on talking about MCAT strategies, and I'll specifically be talking about how to practice with biology and biochemistry passages. So if you want, you can go there. We'll talk about strategies, how to practice. Yeah, so if you have any questions at all, you can email me at ashleymcat101@gmail.com. I check my email always, so my response time is pretty fast. And you can also donate to our GoFundMe because we're trying to cover costs for advertising on our website so that we can reach out to more students. Do you have any questions about what we talked about or anything?
Based on what we talked about, I think I'm good.
Okay, that's good. So did you find this helpful?
It was really helpful. Thank you so much, Ashley.
Okay, thank you for coming. Let me stop recording. Stop sharing.